can travel there together to enjoy and interact together. Over the whole, are you ready to help me there in Lanzhou?
Yeah, every day we are waiting. We have waited for 365 days.
Yes. Yes.
Every day we are looking for
I think it's time to go back, right? It's time to go back to visit each other both ways, okay? 泰国 and 中国, 泰国, 中国 together.
Okay, start.
Yes, I can. Yes, sure. Yes.
Okay, please.
Yes. Yes. 早上好 สวัสดีครับ First of all, I'd like to express my sincere thanks and honor to Professor Ho Fujian and the university. Yes.
Professor Ram will give a brief introduction.
Okay, yeah, I need the introduction. I need the introduction. Just a little bit.
Yeah, hello, Professor Wanapat. First, I will make a short introduction of you to all of us, and in both Chinese and English. As well, we have not been international students here, so I firstly will introduce you in Chinese.
But now all the Chinese are very good at English.
Yeah, you surprised me. Your Chinese are really good.
Oh,谢谢你
Okay, let's begin. It's my great honor to be the host of this section of today of New Year's Symposium of Grassland Livestock Production Systems. I highly appreciate Professor Fujian Ho giving me this great opportunity to communicate with you all. So today, our first presentation will be given by Professor Methan Wanapat from Peking University in Thailand. So I will give a short introduction about Professor Wanapat in Chinese first.
Okay.
Yeah. Wanapat 教授，他是泰国孔金大学的教授，也受聘为我们兰州大学崔颖讲席教授，是世界著名的动物营养学家。Wanapat 教授呢，他毕业于美国北达科他州立大学，然后在诺威农业大学从事了一些博士后研究，他熟练地掌握了7国语言。刚刚大家也看到了，就是他的中文也非常好。Wanapat 教授呢，他主要从事反刍动物营养的教学研究工作。它的主要研究领域包括区域化饲料资源加工调制与反刍家畜高效利用，反刍动物牛胃微生态、牛胃发酵与甲烷减排，以及利用植物吃生代谢产物调控牛胃发酵、提高家畜生产性能等研究领域。目前呢 ，Wanapat 教授已经在就是国际学术期刊上发表了250余篇的SI 论文，获得授权了发明专利24项。并且应邀在国际学术会议报告中做了30余次的大会报告。那么Wanapat 教授呢，他曾经担任泰国孔金大学热带饲草资源研究与发展中心的主任，2000年获得了泰国的最高排名的一个 PC 11级教授，2003年获得了白象最高骑士大十字勋章，这是泰国的一个皇家水袋的荣誉。2018年3月呢，他作为云岭高端外国专家被云南省草地动物科学研究院引进，并获得了由云南省外国专家局签发的首张外国高端人才确认函，确定为云岭专家，云岭英才计划云岭高端外国专家。2019年获得了云南省人民政府的彩云奖。同时 ，Wanapat 教授也先后担任了国际动物农业发展和人类资源机构的高级顾问，同时也是世界粮农组织的专家、中国农业部外专局专家、中国农科院外聘专家，以及云南省的云岭高端外聘专家等40余项的人才称号和兼职。同时呢，他也担任了包括 Asian Australian Journal of Animal Science British Society of Animal Science 等20个杂志的编委。Okay, I will introduce you in English shortly. So, Professor Wanapat has been appointed as the Training Chair Professor of Lanzhou University and is a famous animal nutritionist worldwide. Professor Wanapat obtained his PhD from North Dakota State University of the United States of America and later engaged in postdoctoral research at the Norwegian Agriculture University. He is proficient in 7 languages. Professor Wanapat is mainly engaged in the research and the teaching of ruminant nutrition, including regionalized feed resources processing and efficient utilization of ruminant livestock, ruminant microecology, rumen fermentation, and methane mitigation. Use of plant secondary metabolites to regulate rumen fermentation to improve livestock production performance. Professor Wanapat has published more than 250 papers in international journals, authorized 24 invention patents, and has been invited to give more than 30 speeches at international academic conferences. So, this is the introduction for Professor Wanapat, and let's welcome Professor Wanapat to give us a presentation today. Thank you.
谢谢你，早上好，我非常高兴见到你。今天天气好了，兰州非常好。Thank you so much, Professor Tao Ran, for a very nice introduction. Actually, I have known many people over there at Lanzhou University. So, introduction is not necessarily required. But anyway, thanks a lot for giving a short time to introduce. So today is very special because Professor Hu Jian, under his supervision today, has kindly organized very important New Year symposium of the grassland livestock production. And I'm honored to be the first one of today, right? Because there are other speakers coming from New Zealand and the United Kingdom, and I'm the first one to have a kicked off, right? So it is a great pleasure and honor to see the young scientists, especially over there at Lanzhou University, doing graduate programs. And I am pretty sure that getting together here will be interactive and, with a good discussion and also with the onward recommendations and take-home message for all you guys there to think more and work more and harder, okay? So let me share the screen for you.
Yeah.
I will share the screen, right?
Yeah.
Just, as I said that I have known.
We can see your screen now.
Okay, very good. Yeah, as I said that I have known Professor Hao for a long time and many other people. And during the last 6 to 7 years, I have been traveling back and forth to Lanzhou University to meet many people and learn a lot from you guys there. But only this time because of the pandemic of COVID-19 that has taken us away from each other. But I believe that in the short while we should be able to return and interact with each other. Okay, so just keep in mind and be patient that we believe that we will come together very, very soon. Okay. The title I'd like to share with you guys today is Methane Emission from the Enteric Fermentation of Ruminant Production and Potential Mitigation Approaches, where I am at Conceptoscar University. Okay, just a little bit here. I'd like to thank my student, my PhD student, postdoc, who have helped a lot in preparing the PowerPoint for the presentation. And she Dr. Beer, the postdoc, just just gave me this one. He said, Professor, now you have discovered H index of 35, and that has been informed by a former student at Metro University, Dr. Subban. By the way, today there are other participants from 4 universities in Thailand who are joining with us today. I think it's a good chance that we can interact, and that will take us, for the future to move together more and more. Okay. Just a little bit first. It doesn't move. Just a second, please. Oh, here we go. Just wait a minute. It's a little bit. Okay, here we go. Okay, just to show you guys that Professor Charles Darwin has been saying it very truly that it's not the strongest species that can survive, nor the most intelligent, but the most responsive people or species to the changes can best survive in this situation under this dynamics of COVID-19. Okay, so you are the good group and you have been adapting yourself to the environment. I think the internet is a little bit slow. Here we go. All right, may I just bring you guys back a little bit to the COP26 that was organized in the UK just very recently, and many organizations and representatives were there, and it was very interesting, right? If you follow, the discussions and recommendations, you could see particularly the FAO has set up the 4 pillars that will be very good for us to follow. FAO of United Nations has set up PNL, which has stated very well that we have to improve ourselves for better production, better production of livestock in terms of quantity and quality. And then better nutrition, not only the yield, but, we should have, good quality of the products, especially for the human health. And then onward to better environment. Okay, can you hear me?
Yes. Yeah.
Okay, better environment, especially to reduce the global warming, or to reduce methane production and emission. And the last was for the betterment of livelihood, for all stakeholders who engage in the production to have better life, with better food, with better environment, with better economics, So all of these 4 pillars should be seriously taken and implemented. It's very clearly stated by FAO that we should be aware of. Now, coming back to the global population, it's very clearly shown that there will be a big increase in the global population. So that means that especially the food demand for the consumption should be well prepared for the global population, especially the people who are close to us, like in Thailand, in China, in other countries. We should be able and be powered to prepare the food for them, especially the livestock products, protein products, right? Here, looking at this chart, you will see that all the meat products as well as eggs will be substantially increased from the year from now to the year 2050, starting from beef, mutton, pork, poultry, as well as eggs. You will see that the tonnage of yield of animal products will be significantly improved for the great demand for the consumption of the global population. So we need to be well prepared to do a better job in producing these products to meet the demand. I'm clicking my PowerPoint, but it's moving very slowly. So. It doesn't move. This is it takes sometimes to move it. Now it's just a moment. Oh, now it's going too quickly. Just a second, please.
Take your time, Professor.
Yes, it's internet. You can see the pictures of the livestock production that have been engaged by the global population, starting from like using them as draft, as companionship, as friends, socioeconomic status and all that. And importantly is for food consumption, as I stated that for the protein products, from the meat, milk, and eggs, right? So it's very important for the global population, especially the livelihood, of these animal products that have been serving and supporting the livelihood of the people. That's why it's very complicated to talk about livestock systems. That it's not only to see the products, right? But it's all the complexity of the functions that have been, contributed into the population. We cannot just talk about, for example, global warming. We have to relate it to all of these factors that I have just stated that because of the global population have been engaging in the livestock production, especially for their livelihood. Let's move. Okay, now let's come to our system, our livestock production systems. There are many factors that will influence the efficient production systems, but I'd like to show you that one of the very important one is the nutrition and the feeding that will be impacted to the efficient livestock production. Firstly, are those the feed resources available, accounting from the conventional resources to the non-conventional resources. For example, you can see, like Rosa Ho and you guys have been working on pastures and grasses, which have been very important for the production systems of the livestock and to improve their efficient productivity, you can see. But then the whatever is available there, in each locality, particularly the crop residues and industrial byproducts, that should be accounted for, be used as feed resources. Whatever they are holding or they're having, that should be looked at and improved to, especially the nutritive values of the resources for the better utilization of such resources. You know, like for example, you can see for rockfishes, you can see if they're low in nutritive value, especially the crop-based roots such as rice straw or other straw, you can use one of these methods to improve the nutritive values, particularly the food protein and energy concentration of the resources. And onward to the feeding systems, there are variety of feeding systems that we need to fit well with the resources, you know, to support the production potentials, right? So we have to see what is available and what would be the most suitable feeding methods in order to reach the efficiency of the productivity. For example, you can see if they grazing, how can you feed this livestock or the ruminants, right, to be better off? How to supplement, you know, the feed resources if they're required, and onward to a more intensive production system like feed loading, you know, or separate feeding. What kind of resources we have, whether we need to improve their physical and chemical forms of the feed resources or not, and onward to if we were required to use total mixed ration, you know, for better efficiency of the feed resource utilization. So we have to take all of this information and the available technologies together to be able to reach, you know, the most, I would say, like the most efficient production systems of the livestock, you know. And now, as we have been hearing a lot and learning a lot right now, that there have been precision or smart animal production have been recommended, you know, with nutrients identification and also nutrient check up to meet the requirement of the production system of any animal of such production category. Like, you know, if we need to boost it, or we need to increase the rate of production or average dairy gain, or to increase the milk yield, you know, and what kind of feed resources are available there and what is lacking behind. So we really need to check that very carefully in order to correct it and to meet the requirement for the livestock or for the ruminants to produce good products. Okay, so this is very important to be concerned and to be carefully checked and onward into the future supplementations or implementation of the whole feeding system. Let me try to click again here. It's a little bit slow because I think it's the internet system. Sorry about that. Let's see. It's not moving. Okay. Giving you some example. If you have question, also you can raise your hand. You can present your question as well. Okay. I'm trying to go slowly. Okay. Is it okay?
Yeah.
Okay. So if I'm going too fast, please let me know. All right. So to be better utilize these feed resources, may I give you some examples what to do in order to reach our production goals? Okay. The following feed resources or nutrients should be well taken care of or checked and be well implemented. For example, we need to provide the rumen degradable carbohydrate source. What is available there? You have to identify what source you have, right? Like in Thailand, we have cassava chip, you know, and if you can have like wheat bran or rice bran or molasses. whatever is available as the carbohydrate sources, check, and then check whether they have rapidly degradable or slowly degradable carbohydrate. Okay, and then we have to build in other important nutrients, especially the non-protein nitrogen, like from urea or biuret, for example, in order to trap the carbon skeleton that will be provided in the rumen, and that the microbes can take it further, right, to synthesize for microbial protein, okay, right, to produce protein of the microbial cells. And together, very importantly, are the availability in the rumen of sulfur and sodium. These are very important minerals that should be, incorporated into the diet or into the concentrate mixture in order to be available, and the microbes or microorganisms can incorporate all of this to synthesize very essential amino acids, especially those sulfur-containing amino acids, right? To be available in the lower gut for digestion and absorption, to be used in the target tissues. Okay, and not only that, but we have to better improve the efficiency of the products, especially to reduce the gas that is available or produced by the fermentation process, particularly carbon dioxide and methane, which has been shown, to be very important to mitigate or to reduce, to lower it down, right? Meaning that if we can lower it down, we can use this hydrogen to synthesize for other end products, in the rumen. And that end product would be very useful for the host ruminants, to utilize it later to synthesize products in the tissues, like in meat and in milk, with yield and quality, So, and I will elaborate more later on in regards to using phytonutrients like denstanin or saponins, as well as other resources, in order to be efficient. When using all of this together, we'll be able to increase the protein to energy ratio in the rumen, which will result in improving the total growth of fatty acids, which is a very good and important source of energy to providing to the ruminants, right? Especially increasing propionic acid, which is very important here to be used for energy source and also glucogenic compounds that will be required for synthesis of the products, like in milk and also in meat. Okay, and onward to improving the microbial protein synthesis. Here it is important because when we prepare the concentrate supplement, or we check the nutrient concentration, we will check energy and protein, right? So protein will be provided by protein sources, which is very expensive. What I like to say here is that if we were able to upgrade the synthesis of microbial protein in the rumen, we either can maintain the level of protein in the concentrate, or can reduce the level of protein. If we were able to reduce the protein level in the concentrate, it will have an impact onward to lowering the production cost of the feed, which is very important, because it serves as a major part of production cost in the ration, right? So this is very important that many ruminant nutritionists have been trying to increase the microbial protein synthesis in the rumen in order to achieve times that we tend to forget. These resources have been, not very difficult to manage or to implement, but many times that we tended to forget. If we would simply done, I think that we will achieve the more efficiency of production and increase the yield and quality of the products. Okay, click again. Okay, now in the rumen, I have been repeating many times, but it is very important that we need to understand the rumen ecology. The rumen has to be healthy, just like the body or the human being, right? The rumen needs to be healthy to be able to prevent the incidences that may occur in the rumen. And if it occurred in the rumen, it will influence on the health of the ruminants and the productivity of the ruminants. So we need to understand what it is and how it works, right? At the same time, we should be able to reduce the production of the gas, especially the greenhouse gas like methane that has been emitted out, by eructation. This is the issue, the hot issue that has been stated, globally, how we practically, how we can do it simply, we can do it theoretically. It's not important, right? It's not good. But if we can do it very simply and very practically, and that means that the farmers can do it. What I just go straight into it. Farmers should be able to do it. And then if they were able to do it to reduce the methane, and their production should be increased, and that means that their economics should be improved, should be increased. They will be happy. So this is what simple statement and needs to be stated and need to be concerned. All right. So if we were able to understand all of this, I think the production system should be improved. Okay, just an example. The very important factor in the rumen is the nature and the population of the microbiomes or microorganisms who live in the rumen, and they perform their activities, especially to degrade the feed resources in the rumen to provide, fermentation end products to be further absorbed or to be further transported into the lower gut for later degraded and absorbed for utilization in the tissue, in the target tissue. Okay, it's very important. That's why rumen microorganisms has been really stated and has been recommended to be closely studied and implemented. Let's see, it's a little bit slow. Also, it's slow. Again, this one is very important report by Chadre et al. In 2018 that has been published. Okay, we could see that this one has been published in honor of Professor Hungate, right, who has been like the king or the father of rumen microorganisms. As you all know, that it's very important. And I like to to show you that this report, this publication has given very important information about 75 to 80% of the rumen microbes have been identified with their functions, with their enzymes created by each microorganisms, and we are happy to see it, right? And we should take it forward to learn more and to apply it more into our feeding systems, as I have said already. So if I think you should follow this, track it back and see more details of this data, as well as other data. And some of our friends in China also have published very important information. And from Lanzhou University, many researchers have successfully tackled this information as well. So we should learn more about and also apply this knowledge into the feeding systems in order for these microbes to better utilize the substrates and to especially to produce more C3 and to reduce the gas from the fermentation end products, particularly methane production. This one also to show you that at the phylum level, especially the Bacteroides and the Firmicutes, Bacteria and Fibrobacters have been shown for the changes or the shipment due to feeding system, or due to different feed resources or feeding concentration. May I just say that, so we have to see, because this at the phylum and genus and species level, if we were able to identify which species have been important and how they can live in the rumen very happily and then perform well. And we should be able, especially in providing the diets, the feed and the diets, for them to be better, and then we should be able to see the production yielded from the phenomenon. Okay, now back to, as I said, that it is very important to see how the microbes live in the rumen, how they ferment, right? Because the end products, fermentation end products are very important, especially in emitting out the gas, methane gas, especially, that we are concerned of, and we should be responsible in adapting the feed resources to reduce the methane emission. Okay, here again you see all the feed resources coming into the rumen, both raw features and concentrate, as well as supplements, right? When they come into the rumen, the microbes of all types will secrete the enzymes, to degrade the substrates, to degrade the feed resources or substrates, to yield in principle volatile fatty acids, right? Energy sources. And we need to improve the concentration of C3 and to reduce the ratio of C2 to C3, while let's see, improving the microbial protein synthesis, as I have stated earlier, that how can we improve this, by producing volatile fatty acids, there will be available carbon skeleton, and the carbon skeleton will be trapped by the presence of non-protein nitrogen, especially from the quickly degradable nitrogen, like from urea, that will be easily degraded into ammonia. And nitrogen in the form of ammonia will be incorporated together to produce microbial protein. Okay, this is very important. If we have both the turnover rate, okay, and the yield of microbial protein synthesis to be later available in the lower gut, in the small intestine, to be digested and absorbed and transported to the target tissues, like in meat, for meat production and for milk production, right? So, all the tissues, all the tissue, skeleton, muscle, adipose tissue, mammary gland, all the nutrients, okay, apart from volatile fatty acid, which is absorbed in the rumen and transported to the liver, right? To the liver for through the gluconeogenesis, and the microbial protein will be later transported into the small intestine and degraded, absorbed to be used in the target tissue. So it is happening here in the rumen. That's why, Professor Hungate especially has, stated very interestingly and very importantly that we need to understand, the functions, activities of the microbes who live in the rumen, in terms of not only the phylum but down to the genus and species, what's available there, especially as I will, elaborate a little bit more later for like cellulitic bacteria, who are very important, because the idea and the importance behind this was that what would be most, practical, to use it? How would it be? How would it be implemented, easily and practically, as I said, especially by the farmers and by the industry as well, So this is why I will talk more with more details later, right? You can see Hook et al. In the year 2010, right, have proposed this one. The idea behind this phenomenon are as follows. I will start from the rumen pH. Rumen pH is very important, okay? As stated before, the rumen pH should not be too high and not be too low, right? If it's too low here, that especially the microbiomes of using cellulitic resources will be reduced, will be inactive, okay, to degrade the feed resources. So we need to maintain a somewhat good range of rumen pH. We should not let the rumen pH down from like 6.1 or 6.0. That means it will become more acidic, okay, or higher than like 6.8, 0.9, or even 7 to be more alkaline. Okay, there will be some influence on the shift or the change of the rumen, microbiomes that will affect on the fermentation efficiency. Okay, and at the same time, we should be able to trigger and to increase, okay, the fermentation end products, particularly the total volatile fatty acids and the concentration of short chain fatty acids like C3, right? C3, and at the same time, we should be able to trap the hydrogen, as I showed earlier, okay, to produce or to synthesize propionic acid, and then to reduce methane production. Again here, microbial protein synthesis should be improved in order to provide it, in the lower gut, and that is very important when we trace back to protein concentration in the diet. As I said, that if we could produce it more here, then the animals can utilize it more over here. It doesn't have to come from the concentrate or the feed in the ration, right? Okay, and onward, if we can trigger faster rate of rumen turnover or faster rate of passage, okay, which will trigger or improve the dry matter intake, especially the roughages that is very important for ruminants. So these are some examples of the characteristics that should be taken care of and should be tailored down to implement it, to make it happen in the rumen, right? Simply show you how this efficiency and the greenhouse gas are concerned. You can see it over here. This is the rumen fermentation efficiency. Okay, would increase while the greenhouse gas, methane production, especially methane production here, would be in contrast or in reverse, right? So increase of fermentation end product, which means that the methane should be reduced at the same time. We need to increase the efficiency fermentation end products while increasing C3 and also lowering the ratio of C2 to C3 to increase the fermentation efficiency to later make available, these resources for the synthesis of the important production products. So, and especially when you calculate methane for the yield of meat or the yield of average dairy gain of beef, right? And milk production, average dairy gain of beef or average dairy gain of sheep or goats, okay? Similarly, or the milk yield of methane should be improved to the unit of methane produced, as we have known that on a daily basis, small ruminants like sheep will produce about how much? About 40 liters of methane per head per day, right? While the large ruminants will produce like 200 to 400 liters of methane per head per day. So it depends on species and type of ruminants. But at the same time, we need to calculate it out per unit of production and see how this will come out, right? And see how efficient it would be. So we have to do it, be careful and do it thoroughly. And for methane production, it's a biogenic carbon. It's a biogenic methane. Biogenic methane, which means that it's recycled, right? It's recycled. So that's why Professor Ho and students, PhD students and postdocs have been working a lot, right now. And I have been helping some of you guys, in producing, in looking at the experiment and producing the publication. You can see that Xing Chan in the group or not. I hope he is. Anyway, so we have been working together, right, to see how methane can be recycled. And it's important because carbon and methane can be sequestered, could be trapped by the plant, by the grass, right? And then they can produce more yield and good quality, especially pasture. Professor Ho can produce good grass, right? We can trap carbon through photosynthesis, right? and the grass will be consumed by the animal, like by dairy or beef, and will go through the fermentation process, as I have shown you guys that. Okay, and then if there were methane, it will be emitted out into the atmosphere, and that methane is not a long time shelf life, okay, as compared to thermodynamic methane, right? So it would be recycled into the atmosphere anywhere, although it's not quickly recycled back, but it would be recycled into the system anyway. But having said that, it doesn't matter. We don't have to care about that. We need to be careful. We have to reduce it down through the enteric fermentation, okay, in order to be more efficient, to lower it down, not to allow it to be available in the atmosphere, right, through the biogenic carbon cycle, like that, right? So Methodogenesis is very complicated, right? It's a process have to be engaged or by the action of methanogens, which have been available in the rumen, and many groups, many ruminant nutritionists have been interested in working with methanogens and see what's available there and how they're, what would be their ecological characteristics in order to learn how they live, how they utilize their feed resources, and finally how to reduce the methane produced by methanogens, or how to reduce the population of methanogen. It's very important, okay? So it's a process that is complicated, as we have been knowing already. And the work by Professor Atwood in New Zealand have shown you very interesting how methanogen have been working in producing the methane through the reduction processes through various enzymes and coenzymes and cofactors available over there. As you can see, that this work is complicated, and I'm very happy, by the way, that some of you sitting there have been working, I'm sure, on this methanogens diversity in the rumen, in the yaks, right, that have been published and maybe ongoing. I think that more work should be done even more to see, in particular in yaks, that how they can better utilize lower quality of fibers. I think it's very important to see how they utilize it, and how the rate of production of hydrogen is very important as well, and how the methanogens can trap it. We have to study the rate of production of hydrogen into the synthesis of methane output. I think that still requires a lot of data to see in terms of the rate of production, and then I think that we can learn and apply even more how to implement other resources in terms of blocking the process, as I shown all of you already, right? Okay, this is just to show the structure of the plant cell walls. You can see that that fiber is quite well demonstrated that it's not easy to degrade the fiber because the lignin is lignified, okay, with the three components, as we all know. But how we can easily utilize it, we have to either improve it, either treat it, or what? I mean, what kind of microbe that will be more efficient than others? That's why we have, many people have been interested in looking at this context. see the enzymes that have been involved, okay, or engaged in the degradation of the fiber. So different groups of enzymes, you know, coming from endocellulose, exocellulose, and finally the beta glucosidase that have been shown by Dr. Prof in Australia from Queensland, that to see how these enzymes can work together and the rate of the enzymes secretion and also the activities, you know, how they behave. This is very important to see how they work. Okay, now back into. I'm just looking at my time now. Okay, I have to hurry, speed up a little bit. Okay, now I think the internet is better now, right? Okay, so you see, pH is very important. As I said, we should not let the pH of the ruminant lower. Otherwise, it will create more incidents and more problems. Okay, you see. Now again, I show the work of Professor Oskov, of the late Oskov, okay, and our work as well, that to see a ratio of roughage to concentrate have greatly influenced, okay, on the pH. You see that higher roughage to concentrate will be able to maintain at a higher pH. But if you lower the fiber, the ratio of fiber down and more concentrate, when you see the pH will be declining, okay, even lower than 6.0 here, especially on the very high concentrate supplementation. This is very dangerous because you see that more acidic condition will be happening, will be creating over here, which will then impact on the instances in the rumen and to the ruminants. Okay, so we have to maintain a good ratio of roughage to concentrate. If we were able to formulate a total mixed ration to see, okay, and at the same time on the influence on the volatile fatty acid production by the late Professor Oskov and our work as well, follow similar trends. Okay, we would at a higher ratio of roughage to concentrate, we have more C2. Okay, if we lower the ratio of roughage to concentrate, the C3 would be also increasing. You will see that. Okay, very importantly, very similar patterns that have been obtained over here. So normally the ratio, if we were able to formulate the total mixed ration or to either ground form or to pelleted, I think that the ratio should not be lower than like 40 to 60. I think if we lower the fiber or roughage from 40 to 60 concentrate, I think that the pH will be dropped dramatically. That will create more problems. Okay. Another work here very interesting that the NDF digestibility per fraction rate, you will see that will be influenced by the changes of pH. You see that if it's lower than 6 over here, so the digestibility will not be increased. Luckily, it could be maintained, right? But in many cases, it will be dropped down like that. So you see, this is pH of 6 over here. So you will see. The pH dropped by after 6. You will see that the rate of the fraction of the NDF digestibility will be dropped. See, you will see that very dramatically. So better we will have to maintain the rumen pH of above 6.1 up. Okay, this is very important one. Now, may I just come back into the weight using the phytonutrients or plant secondary compounds, okay? Because have been interested by many researchers on the use of these resources, okay, either from the plant or from the byproducts. In our groups, okay, we have been interested in employing the product from fruits, okay, fruit peels of wastes, okay, which contain phytonutrients or condensed tannin and saponins. And how these phytonutrients work, for example, may I just bring it up here again, that the phytonutrients will have influence on the cell membrane of the bacteria in the rumen to interfere with the transport of nutrients across, okay, that will be lowered or will be less efficient, right? So there will be less nutrients available for their functions. So the membrane will be destroyed eventually, okay? And also it has been shown that this not only on the cell membrane of bacteria and also protozoa, but also on the other functions like fractula of the bacteria, that is very important to perform the attachment. Okay, when the bacteria will degrade their feed resources, they will attach to the surface of the feed resources, but these phytonutrients will interfere with the fractula that will influence on the blocking or the blockage of the attachment to the feed substrates, which will then lower the degradation activities and fermentation end products will be reduced, okay? And we have also, yes, and also the influence on the protozoa in particular, that the phytonutrients will have the ability to bind to the storal moiety or to the storal moiety of the cell membrane in particular, you know, and then the protozoa membranes, okay, will be deteriorated, will be reduced, and then you know, the ability to utilize the nutrient will be less, and finally, methanogens which have been living or attaching to the surface of the protozoa will also be destroyed as well. This is why we have all the result have been found or referred to was the reduction of protozoa population, and as well as methanogen have been found by using these phytonutrients which contain, by using these food peels which contain phytonutrients, or plant which contain phytonutrients that have been incorporated into the diet or into the concentrate, you know, if you were able to use it like that. Under our work, we were also interesting to find that Fibrobacter succinogenes, which is very important bacteria available in the rumen, and Fibrobacter succinogene is considered to be the non-hydrogen producing bacteria, has been found increase, you know, when we implemented the food peels which contain phytonutrient, which is very, very important in a way to explain that why methane production has been reduced, and also propionic acid production has been increased. Under our work, we have been using the product of banana, okay, to mix a product called, this is the name Bioflop, and use it to manipulate the rumen pH, right? You can see this work we have tried to supplement or Bioflop at various level of supplementation from 0 to 3% of dry matter, and we sampled and checked the pH. As you can see from hour 0 to hour 6, you can see that 1% of dry matter intake. After that, increasing the level of supplementation of Bioflop, will upgrade or will improve the rumen pH. You can see that, higher level, 2 to 3%, were able to perform more respectively of hours. You can see it. The average you can see over here from 0, 1, 2, and 3% of dry matter. So Bioflop, which is a product of banana, which contains phytonutrients, particularly condensed tannin. Okay. Now you see this work show you also that using different fruit peels which contain condensed tannin and saponins, from mangosteen peel powder at 100 grams and banana flower powder at 100 grams, and the combination. And you can also see that the influence on the production of total volatile fatty acid and the production of C3 and the ratio of C2 to C3. And finally, influence on the production of methane by using either source of fruit peel powder, and the combination would have influence significantly on the production of C3 and the methane reduction. You can see over here. All right, and onward to other experiment by using different types of phytonutrients from the fruit peel. You can see that here we have found total volatile fatty acids and C3 propionic acid were significantly improved, where C2 and methane production were decreased by the supplementation of fruit peels at 100 grams per head per day. So this is interesting to see how simple it is, to implement this. Okay. Now with dairy cows, we will also found the significant increase of using the fruit peel powders, in the supplementation. This is milk yield and the 3.5% fat corrected milk by using fruit peel of rambutan peels powder, and the combination of urea will further improved the production of 3.5% fat corrected milk, as well as you can see because of the fat increase, so the 3.5% fat corrected milk will eventually significantly improve as well. Interestingly, total solids will also improve. So under this trial, let's go and see other work. This is a little bit by other sources. This one was done quite a while ago, that we were interested in using the lemongrass powder which contains essential oil. You will see the changes of different rumen microbes, starting from bacteria, protozoa, fungi, zoospore, and other types. You can see that this supplementation of lemongrass powder here from that we have, usually is we use in cooking, right? Lemongrass powder over here, but we can implement it in the concentrate supplements as a supplement that will influence on the population of the rumen microbe with different levels of supplementation. You can see that this is bacteria, protozoa, fungi source, but especially on protozoa, you see very significantly reduced, of the protozoa population. All right. Again, this one is also lemongrass from the population of bacteria, protozoa, and fungozoa spores. Okay, let's move on now to another very recent product. A very recent product that we have developed, we call Prof. And the most recent one is Marabac. This is a product formed in the form of pellet containing fruit peels. Okay, what sources that have been used? Okay, I will show you here that the sources from mangosteen fruit. This is the mangosteen peel powder of 45%, which is a major proportion of the pellet. Okay, mixed with other kinds of fruit like rambutan or banana. Okay, mixture together to form the Marabac, for example, over here, which contains about 9.5% good protein and various level of minerals. Interestingly, that the mineral from the fruit peel also, can be protein, particularly the two sources, the condensed tannin and saponin, were combined together, were found, in the Marabac product, and we use this one in quite a number of trials from using in vitro and in vivo as well. Okay, you see the effect of Marabac, for example, here, that will result in improvement of maintaining or uplifting the rumen pH and improving the rumen fermentation. Okay, right. And as I said before, that we were able to obtain good population of Fibrobacter succinogene, which is a non-hydrogen producing bacteria in our trial, which was very nice, to obtain such data, right? Although this data is not quite sufficient yet, we need to do more work on tackling, the instance of having more Fibrobacter succinogene in such ecology. How, so, and also improvement of C3 and lowering the ratio of C2 C3, increase nitrogen balance, increase protein, microbial protein synthesis, improve the digestibility of nutrients, and increase the production of milk yield, and milk yield per unit of methane output. At the same time, we were able to reduce protozoal population and less methanogens, lower ammonia nitrogen. Lower ammonia nitrogen could, may I just add a little bit here, that it could have been due to the effect of condensed tannin binding to the protein in the rumen, or to increase the rumen bypass protein into the lower gut. So so the protein resources could have been less degraded in the rumen. Therefore, ammonia and nitrogen concentration were produced less, right? So this could have been the effect of the tannins binding effect to the protein, and eventually having less methane production in the rumen and less methane emitted out into the atmosphere. Some of the details of the work can also be seen here, that after hours of sampling, 24 hours sampling, we'll see the supplementation, okay, at low level of concentrate supplementation and a high level of concentrate supplementation will be able to increase the pH after hours, after respective hours of sampling up to 24 hours. You will see like at 24 hours here, this is at a low level of supplementation of concentrate. Still, the supplementation of Marabac would upgrade the pH. This is at a high level of concentrate supplementation, whereby the rumen pH would be reduced, right? So supplementation of the Marabac then could lift the pH up. So this is why pH is very important, you know, when we supplement it with the phytonutrient pellet like this. Okay, look at the fermentation end product C3 and the ratio C2 to C3 as well as methane production were significantly affected by the supplementation, both at the low at the high level of supplementation. At the same time, the level of supplementation of 200 grams per head per day were used under this trial. Okay, in overall production, we were able to conclude it or to summarize as follows. Using Marabac pellet, okay, as a supplement which contain phytonutrient, we'll be able to upgrade the pH. You can see over here the pH dropped down and the pH were increased. Okay, and protozoa population were reduced. Okay, this is, and methanogen of course will also reduce. And importantly, the concentration of C3 glucogenic compounds concentration has been significantly improved or increased. Hence, the methane mitigation was achieved by using the supplementation of Marabac of containing these phytonutrients. Some other experiment, you know, could be summarized in the next two frames of PowerPoint here. You can see, and I'll show you the publications that we have published as well. This is in vivo trial. You can see some of other work under our groups. Okay, under our groups, there have been other researchers, like this one is Chert Tong, okay, who's a young, very potential researcher in our group here, has conducted numbers of trials with his graduate students and have found very interesting work on reducing methane production and emission. You will see that through the reduction of protozoa and similarly through the support of increase of C3 and digestibility. Okay, so you can see over here onward to other trials. Quite a number of trials have been conducted. You see, using different sources, okay, that are available in our area. So in other countries, in China, in Lanzhou, I'm sure there are other sources that could be available over there, and you can identify and see the concentration of phytonutrients in those resources, and could be studied in terms of concentration, how it could be formulated, you know, to make either to mix as a powder or onward to make it as a pellet and use it in the supplementation. Okay, so onward to other trials like Marabac, as I mentioned, from different sources. Particularly this one is from dragon fruit peel, and I'm sure you know this one as well. This paper has just been published over here that he used in. This one has previously published in the dairy bulls, but next trial of his was also just recently published, just last week actually. I'll show you the journals as well. Very interestingly to see, okay, and this one just to show you a little bit that okay, with the animal products, okay, meat, milk can contain lots of bioactive compounds, not only concentration of protein and amino acids, but there are other bioactive compounds available in the animal-based products, which is very useful for human consumption, for their health, okay, and for their well-being. So we will discuss about this functional role of the protein, animal-based protein products more later on in other occasions, okay, when we have more time. So I think that because the public has been talking a lot about protein sources from other sources like plant-based protein or from insect protein-based, things like that. But I would encourage many people to carefully see the quality of the protein products, protein-based from animal-based protein, okay, available, to see how useful they are and how we can also manipulate it in terms of improvement or effectively utilize these bioactive compounds, I think. And then more than that is also the engagement and association with the livelihood of the stakeholders are utmost importance. Okay, see, this is a very recent paper, okay, by one of my PhD students, okay, just published in Animal Nutrition. Okay, he used this dragon fruit peel in dairy cattle. I like to invite you guys to check the details, how these results have been found in his paper, just published actually just a few days ago, okay, in the Animal Nutrition Journal, which is based in China and ranked very highly, okay, with very high impact factor, right? Now Lanzhou University, yes, I'm very impressed, okay? Let me just say that I have been many times with my good friend Busa Hao over there, and many people there, 1909, and it's very, very old university and with many good work already, so you guys have done great work as well in this research here. Let's see who are here together. Professor Sadruddin was with me, my wife, and she was over there. She's a plant ecologist, and she was happy to present the work over there at that time. And Professor Hall, she said that she wished to go back again. Anyway, to visit you guys and give a special lecture to all the students over there. So you see our students over there, they look at our students. We were together from the beginning, like 6-7 years ago, at least. I was there happily talking, sharing with the student, So it's very nice to interact like that. I think we need to work more. See, this is me again, and with all you guys over here, maybe graduated already. Maybe not. Maybe not. Where's Ching Chan? Ching Chan is not here. Anyway, so all the young students working very hard, and all of you guys there. You say Ching Chan is here, okay, and other students send all my best.